Good morning students. Today I am going to teach about preparation of herbarium specimens. So I have just shown some pictures how a herbarium sheet will look like. So what is a herbarium? So why should we go for preparation of herbarium sheets? So herbarium is collection of plants that are being dried, pressed, preserved on herbarium sheets and arranged according to any accepted system of classification for further reference and studies. So you would have studied lot of systems of classification like Bentham and Hooker classification, Hutchinson classification. Normally in a herbaria they use only Bentham and Hooker system of classification. So what are all the steps for preparation of herbarium specimens? So this semester you have taxonomy of angiosperms, where you are going to study about how to classify angiosperms, what are all the morphological features of angiosperms with which you can identify to which family a plant belongs. So why should we do this herbarium specimen? If you have this herbarium specimen only, you can find out what a plant looks like, or actually what is the shape of the plant, how a flower of a particular plant will look like. So there are few steps, I will explain one by one. The first step is field visit, collection of specimens, pressing of the specimens, drying of the specimens, mounting them on a herbarium sheet, labeling, and you have to properly store them in a system of classification. First and foremost step is field visit of specimen collection. So why should we collect? What we have to actually collect for herbarium? It depends on whatever topic you are dealing with. Suppose you need to study about the flora of a particular area, we need to collect all the algal specimens, fungal specimen, bryophytes, pteridophytes, gymnosperms, lichens and angiosperms. So if you are going to prepare a herbarium specimen only for angiosperm, in that case we have to collect only the angiosperm specimens. How to collect a specimen? So there are different types of habitat is present, like for example you have trees, you have shrubs, you have herbs, you have climbers. So in case of trees, you can't cut the entire tree. There we need to go for collecting a small twig alone. In case of shrub also we can collect a twig, preferably they should have flowers. If not, we have to collect them with fruits. In case of herbs, we have to collect the whole plant, which means you have to dig them out from the soil along with the root system. So how many specimens should we collect? So normally when we take the girls for a field visit, all of a sudden when they see a flower, they go and collect all the flowers. That should not be the case. We have a social responsibility. We need to preserve the biodiversity. So we can collect 2 or 3 specimens if we are going to submit as a herbarium sheet. In case of flora studies, we have to collect 1 type specimen and followed by that 3 more specimens we can add upon. Why should we go for regular field visits? What is the purpose for regular field visits? Like when you are doing floristic studies, you may have the flowering season in 1 month and fruiting season in 1 month. Suppose you go in the month of June, you see the flowers So you don't get the fruits, but when you want to collect the fruits, you have to go for 1 more field visit in the month of August or September, depending about the fruiting month. So these are all the things that you have to follow when you go for a field visit. So next, when we go for a field visit, we need to carry some tools. What are all the tools that we need to carry? So the first figure that I have given is a small knife, which is a pocket knife. You can have it. It's very handy. Or if you are going to go for a big group and you need to go for collection of several trees, then you can take a big knife. This is the figure B is scissors, or you can say it as a cicature. You need the cicature to collect big twigs. And in case you are going to collect a shrub or you are going to collect a herb, you need a digger so that you can dig out the whole plant along with the root. Followed by that, sometimes the field visit may last from morning till evening. So in that case, you can collect all the specimens and you can put it in the vasculum. D figure shows vasculum, where you can just dust out all the dust particles present in it and the sand particles, and you can put all your plant specimens inside the vasculum, which will make the specimen fresh till evening. E is plant press and F is herbarium sheet, which I will teach in next slide. So these are the actual pictures where the vasculum is present. You have the photograph of cicatures. 3 different types of cicatures are there. You can use the first and second one for tree specimen and the third one for shrubs. Next is wooden plant press, where you can keep the pressed collected plants in the newspaper, and you can just fold them and keep them under this wooden press, and daily you can just screw them so that it becomes very tight and it is dried up very easily. So when you go for a field trip, what are all the materials which you should carry and which you should not carry? Because we are going to go into the forest patch, we should not carry any plastic wrappers with us or any food items that are being wrapped in plastic. Maybe you can take a food materials wrapped up in banana leaves, but not in a plastic leaf, because after eating we tend to put all the plastic wrappers in the forest area, which is restricted. You can't take nylon ropes. Instead of nylon ropes, you can take jute. So what are all the things that you should carry along with you? We need newspapers in case we find out many plants and we have some time to press in the field area itself. You can collect the plants and press them inside the newspapers. And in case you want to identify any plants that you have collected immediately, you should have a manual or any flora. Suppose I go to Chengalpet for a field visit, I can take Chengalpet flora or Madras flora along with that. You should always have a field notebook with you to note down what are all the features of the plants that we are collecting. It is preferable to use a pencil rather than use a pen. In case you want to press any flowers or you just want to preserve any fruits, you can take ethanol along with you in small glass bottles. And you should also have a tag so that you can just tag the plants then and there when you collect them from the soil or from the trees. So next is a very important process, that is pressing and drying of the plant materials. So far we have collected all the specimens and we should give more importance in the pressing aspect. Normally we can use newspapers, old newspapers or blotting sheet to observe all the moisture content from the specimens which we have collected. You should not keep the specimens like I have showed in the figure 1, where it is all clustered and clumped together. You need to separate all the specimens with a handle, or you can just use a needle and tease out all the specimens. They can be folded in the shape N or W. So the blotting sheets with plant specimens, they have to be placed in the plant press for drying, and after 24 to 48 hours the press can be opened. So this is a press made up of a wooden plywood, where there are 2 plugs, an upper part and the lower part. You can keep all your plant specimens. So when you keep that, you have to keep only 1 plant per 1 newspaper. So you have to pile them one above the other, and you can keep the second plywood and tie them with the means of jute thread. Okay. So generally the plant should be pressed flat. To get good specimens, the plant must be pressed before wilting. So once you collect it, you can go to your place of stay and immediately that evening you can just press them. The plants, if it is going to wither very easily, the plants which have compound leaves normally wither very easily, or small leaflets, in that case you can preserve them in the tin vasculum, and after the trip you can come back to your room and press them. To get the finest specimens, the plants must be pressed directly in the plant press then and there as soon as they are collected. Some students have the tendency to collect them in a plastic wrapper and spray water. These practices should be avoided. So when you just press the specimen, this figure A is wrong. We should not overlap the leaves one above the others, but the leaves have to be separated using a needle, sharp needle. If it's going to be a compound leaf, the leaf should not be overlapped. In case there is overlapping, you can just cut off the leaves, leaving some portion of the petiole to show what is the arrangement of leaves. Next process is this pressing and drying process can take place for 15 days. In some cases, depending on the season, it can also last even for 1 month. Once you see that all your specimens are dry and no moisture content is there, then you can go for mounting. The first figure shows the herbarium sheet. This is the herbarium sheet. It has a size of 41 into 29 centimeter. Mounting has to be done with the help of glue, and adhesive and sellotape should not be used. Instead, thread can be used for attaching the stem portion to the sheets. In case of pressing the herbarium, we should not press the bulky plant parts like dry fruits, cones. They can be dried without pressing, and they can be put in a small envelope covers called as fragment packets, and they can be attached along with the herbarium sheets. Next important part is labeling. Without labeling, a herbarium sheet has got no value. A label is pasted or printed on the lower right hand corner. The label should indicate the information about the locality, altitude, habit, date, time of collection, name of the collector, common name and complete scientific name. Complete scientific name is a binomial name, and you have to give the common name. You can approach the local people from where you have collected, and you can ask them the local name. Date of collection is very important, so that we can know whether the plant is in the flowering stage or in the fruiting stage. We must write down the family name and the locality, and altitude is very important. We should also note down whether the habit is a tree or a shrub or herb. You can also note down some of the uses if you get the information. Once the herbarium sheet is ready, it is dried, pressed, and it is affixed to the herbarium sheet. The last procedure is storage of the herbarium sheets according to the system of classification. So storage is properly dried, pressed and identified plant specimens which are placed in a thin paper folds. You can see the thin paper folds. They are called as specimen covers. They are kept together in a thick paper folders, which is genus cover, and finally they are incorporated into the herbarium cupboards in their proper position according to the Bentham and Hooker system of classification. You can see small shelves. Each and every shelf is called as a pigeonhole. So what all things can be preserved or it can be collected apart from angiosperms? You can collect and preserve algae, fungi, lichens, bryophyte, ferns, other types of pteridophytes and gymnosperms. In case of algae, you have filamentous algae and macrophytic algae. They can be collected from the natural habitats, and they can be collected using net or directly by hand using scalpel. After collection of different algae, they are cleaned with the water from which they are taken. If it is river water, they have to be cleaned with river water. If it is going to be marine water, they have to be cleaned with marine water, and then they can be finally pressed in between butter papers, and when dry, they can be affixed on the herbarium sheet along with the butter paper to the herbarium sheet. This is the diagram where you have the pedina, and all the specimens are labeled properly for their identity like we do in the angiosperm. So marine macroscopic seaweeds can be collected, dried and pressed like this. So next we will see how the fungi can be collected and preserved. So aquatic saprophytic fungi are collected along with the substratum in which they grow, and they can be preserved in formalin as such in small bottles. In case of Ascomycetes, Basidiomycetes fungus, you have a conspicuous fruit bodies. You can collect those fruit bodies and they can be preserved either in FA or 5% formalin, or they can also be dried and preserved as I have shown in the diagram. Next is lichens. We know that lichens grow on the surface of bark of tree, or it can be grown on the rock areas and crevices. So lichens are collected from tree blocks or from root substratum using a scalpel, either from the bark, or it can be also collected along with the thalli and dried. The specimens can be preserved as herbarium sheets, or these lichen specimens could be stored in a paper pockets with proper labeling. Next group of plants is bryophytes. So the bryophytes are collected from their natural habitats using scalpel and forceps. Different types of bryophytes are there. Some are liverworts, some are hornwoods and some are mosses. So they are thoroughly washed in water, cleaned properly and dried at 15 degrees centigrade or in the room temperature, and they can also be preserved as herbarium sheets. You can see some of the bryophyte specimens which are being mounted on the herbarium sheets. So next we can go for collection of ferns and other type of pteridophytes. Usually the whole plant along with the rhizomes, stobili or sori is collected for collection, so that sori plays a very important role in the identification of pteridophytes. They can be collected with the help of scalpel and forceps. Fresh specimens are collected from their natural habitat and mostly dried for preparation of herbarium sheets. You can see the pteridophytic collections. Next you have some of the ferns and pteridophytes. In some cases you can separately store the ferns rhizome, fertile and sterile fronds, or else you can also store the whole plant or plant parts, and you can also preserve them as a herbarium sheet also. The plants which are being stored in containers can be preserved using 10% formalin. Collection of gymnosperms. Usually it takes of about 15 to 25 centimeter size, along with the sex organ should be collected from their natural habitats. The specimens are pressed and dried for herbarium. Young twigs or cones can be collected and preserved in 10% formalin for anatomical studies. Dry coniferous cones, sporophylls of cycads and jingo can also be collected from their natural habitat and preserved as dry specimens. So after collecting all the herbarium sheets, it is very important that the herbarium sheets have to be preserved for a very longer duration. So these plants, sometimes what happens is they may be infected by some kinds of insects. So insecticides like para-dichlorobenzene, which is called as PDB, carbon tetrachloride or DDT can be used to prevent the infection caused by insects. Next is fumigation, which is made by storing home or cabinets from time to time, where a container is taken and certain fumigants are added to the container and kept in a room, and inside the room all the specimens are being piled up together and the room is locked for 24 hours. After 24 hours the room is open, windows are open so that the gas fumes goes outside, and then we can take back the specimens and put them in their cabinets. Sometimes you can also do the heat treatment where the mounted sheets are being placed in a suitably designed steel almirahs for 4 to 6 hours at 60 degrees centigrade, where all the insects and their eggs are being destroyed. The mold fungi also do sufficient harm to the plant specimens, and if they are not well dressed by some effective chemicals, they may cause a huge amount of damage. For this purpose, the plant specimens are sprayed with 2% solution of mercury chloride, which is highly poisonous. Care has to be taken when we use this chemical. We can also use mothballs, naphthalene flakes in shells of herbarium to check the insects then and there. They act as repellents to insects. So preservation of type specimen. So what is a type specimen? When we designate a particular genus, the specimen which is being collected is called as type specimen. For example, you have Bignonaceae. Bignonia is a type specimen which is very important. So these specimens are those which are used by the author, and it's very valuable, and it has to be preserved with separate shelves with special care. They have to be handled with care and kept away from regular specimens in separate steel cases by the herbarium curators. Usually the type specimens are kept in separate envelopes. So we have studied how a plant can be collected and preserved into a herbarium sheet, how they have to be stored, how the herbarium sheets have to be preserved for a very long time. So they have to be kept in a herbarium. Let me give you 2 or 3 herbarias which are present in our country. The Herbaria of Agriculture College and Research Institute is seen in Coimbatore. National Botanical Garden Lucknow also has a herbarium with about 200,000 to 40,000 specimens respectively. There are about 25,000 specimens in the herbarium divisions of mycology and plant pathology at Indian Agriculture Research Institute, New Delhi. The herbarium of the division of botany at IARI, New Delhi contains 3,000 specimens. So why should we have this herbaria? What is the purpose? The drawings, photographs and descriptions of the plant cannot show the details of structure and development for the students. Usually visual trait is very important so that the appearance of the plants will be remembered by the students for a long time. So herbarium accepts this purpose. Next is comparison of description with the actual specimens from which we have collected from the field. The third point is comparative studies for taxonomical purpose when we do the floristic studies. Plant collections are much used for comparison with new herbarium sheets when new materials are collected from different localities. So what is the role of herbaria in modern taxonomic research? So herbaria and herbarium taxonomists have contributed so significantly to our understanding of natural history, and there is no need to defend their existence. So herbaria plays a major role when any taxonomist does studies pertaining to a plants which have been collected in 1900s. So for that herbaria serves as a major pillar. There is also an urgent need to undertake extensive intensive explorations of all parts of the country. So you can go and see all the herbarium specimens which were collected in 1950s also. This is possible and could be carried out very effectively if the universities start taking an interest in developing herbarium. So each and every college and university should have a herbarium so that the students who are studying in the college can visit the herbarium and know about the plants that are present in our college. We also have one herbaria in our college, especially in the plant biology and plant biology department, where we have nearly 200 sheets of herbarium being preserved and stored. Thank you, girls.